Hi, Year 12s. I'm just starting us off with a PowerPoint that covers all of the topic of cellular respiration. Now, some of this we did go through in class, but some of it we had to rush through towards the end before school finished a bit earlier than planned. So I've just put it all here in this PowerPoint, and I'll discuss some of the main points as we go through it. Obviously, this should be used in conjunction with your EdRolo textbook and your EdRolo online videos. Just a couple of things on the narrated PowerPoints. If you haven't really used them before, at the bottom left part of your screen, you should see a few little icons. They let you go between the slides, but also there's one that has three little dots. And if you click on that, it gives you the option to pause the speaking. And sometimes I will ask you to do that when I want you to have a look at a question or a look at a picture or a graph and have a think about it. So it might be worth going down and just having a little play around with those icons, but particularly the one where you can pause and then start again. Just note if you press the space bar, it will change to the next slide. So maybe avoid doing that in case you miss some of the information. On this slide is all the information in the VCAR study design on what we need to do in this topic. So we talked a bit about the purpose of cellular respiration, and we started looking at the inputs and outputs of the stages, as well as our equations. This we did kind of fly through, so that's what we'll go through properly in this session. I would recommend doing this session maybe in two sittings. It's a lot of information. We probably would have spent a whole week, so probably two doubles and a single getting through this in class. So maybe look at allocating that amount of time. So the purpose of cellular respiration is to convert nutrients into ATP, into energy. So we looked at photosynthesis and how that makes glucose, and now we're breaking down that glucose and converting it into energy. This is a process that occurs in all living cells. Okay, all cells will go through some version of respiration, but the types of respiration are a bit different depending on where that organism lives or what kind of organism it is. So we have our aerobic respiration, and that is when there is oxygen present. Most of what we focus on is aerobic respiration, okay? This produces much more ATP. It's a more efficient process in making ATP. So that is aerobic respiration. I like to remember it like aero, like the chocolate bar, has those bubbles in it. So aerobic is when there is oxygen. The other type of respiration is anaerobic. Okay, so the opposite, when there is no oxygen available. And this produces much less ATP. So usually this happens either if an organism lives in an environment that has no oxygen, or if an organism doesn't have any oxygen available. And we'll look at some examples of that as well. So the process is a catabolic process because we're breaking something down. So remembering in photosynthesis, that is anabolic because we build a glucose molecule, whereas this is catabolic. We're breaking down that glucose, and it is exergonic because it releases energy. Whereas again, photosynthesis was endergonic because it requires energy. We'll come back to this picture a few times. As you know, I think it's really important to be able to see the big picture of anything we're looking at. And this is that relationship between photosynthesis, which we've covered, and respiration over here. So in photosynthesis, the main thing that gets made is the glucose. And also we have oxygen released as a byproduct. Okay, so remember that in our light dependent stage, oxygen is released, and then at the end, glucose is made. Both of those outputs of photosynthesis become inputs of cell respiration. So the glucose and the oxygen get used in cellular respiration. And our outputs of cellular respiration are water, H2O, and carbon dioxide. And both of those things go into photosynthesis. So it is a beautiful cycle there. The big difference really is the energy source. So remember, in photosynthesis, light energy from the sun is captured, whereas in respiration, energy is produced in the form of ATP. Okay, so that is a big difference. But the glucose coming into cellular respiration, the water and CO2 coming out is a fairly standard pattern. Now our equations. We had our photosynthesis equation. With respiration, we have a few different equations because we have one for aerobic respiration and then we have two different equations for anaerobic. The main one that you will be required to know is the aerobic one. You do need to know the others as well, but generally there's more focus on aerobic respiration in the exam, but it might not be the case always. So aerobic respiration, if you remember back to our picture before, we had glucose and oxygen going in, and we had water and carbon dioxide coming out. So you'll see that here, glucose in and oxygen, carbon dioxide and water, and of course ATP. That is the main output. That's what this process happens for. So that is our word equation. A chemical equation, we have our glucose molecule, so one glucose molecule, 6 oxygen molecules go in, and then the outputs, we have 6 carbon dioxide, 6 water, and 36 to 38 ATP. In the exam in the past, they have accepted 36 or 38, but generally we recommend writing 36 to 38 because there is some slight variation in that. So that is for aerobic respiration. Anaerobic respiration is a little bit different, as I mentioned, in animals compared to in plants and yeasts. So in animals, the big difference you notice here, we don't have the oxygen coming in because this is anaerobic respiration. So we just put glucose. Glucose is the main input, and the outputs are lactic acid and 2 ATP. So if we compare that to 36 to 38 ATP, it is a much less efficient process. So in animals, including in us, in humans, glucose goes in and lactic acid and a small amount of ATP come out. In plants and yeasts, glucose goes in, again, still no oxygen because it is anaerobic respiration. Glucose is the input, and we get byproducts of ethanol, which is alcohol. So ethanol, carbon dioxide, and again, only 2 ATP. So both of these processes are much less efficient at producing energy than aerobic respiration. In our last session before we broke up, we did a mind map. I printed out a mind map for you, and this is a small summary of that. Now, I think that is a really useful summary of all of the main processes, but we'll just go through the initial details of that. So you notice at the start, we're looking here at the processes now of cellular respiration, how it all happens. At the start, we have a process called glycolysis. Now, whether it is aerobic or anaerobic respiration, glycolysis always happens first, okay? And we'll go through the steps of it in a second, but glycolysis is always the first step. If oxygen is available, we go through to aerobic respiration. And that will happen in the mitochondria. When we get to this step, we'll have a little bit more of a look at the structure of mitochondria because it is important. But we go through glycolysis, and then if there is oxygen, aerobic respiration happens. If there is no oxygen, anaerobic respiration will happen. Again, we'll look at these steps in more detail, but we also added into our diagram there the 2 types of anaerobic respiration. So there's the lactic acid fermentation, This word fermentation is often used to describe anaerobic respiration. So if you see that, that's what it's referring to. So lactic acid fermentation in animals, or alcohol or ethanol fermentation in plants. So this is really just to give you an overview of what we're going to go through. We're going to start here with the details of glycolysis. So if we actually look at the word glycolysis, and I'm a big fan of using reminders of words or breaking down words. So glyco, like glucose, because basically we're referring to the glucose here. Lysis means to break something, to break a bond. So this is basically the breaking of glucose, this first stage. This happens in the cytosol of a cell. That's important because if you think about prokaryotic cells, they don't have organelles, do they? So prokaryotes don't have mitochondria. They still need to be able to produce some energy. So glycolysis, which is the first stage, occurs in the cytosol of a cell. We have our glucose input, and it's broken into two molecules called pyruvates. Now I just want to note here, over the next few slides, there's a bit of a summary diagram on the right, as well as the inputs and outputs for this particular stage. And on each slide, I have made the inputs green in the words over here. So glucose is green, and I've made the outputs red. Okay, so hopefully that just makes it a bit easier to follow. So in the cytosol, our glucose molecule, remembering our glucose has 6 carbons, it is C6H12O6. So our glucose gets broken into 2 compounds called pyruvates, and each of those has 3 carbons. So if you think here, we've got our glucose, we've got the 6 carbons here. It'll go through this process and we get a 3-carbon pyruvate here and another 3-carbon pyruvate here. During this process, a few things happen. A little bit of energy is released. And we know this energy is produced by combining ADP and phosphate, which we'll get to in a minute. Some hydrogen ions and electrons are also released. So remembering this original glucose is not just 6 carbons, it also has the hydrogen there as well. So when this happens, some hydrogen and electrons are released. The energy is used to combine 2 ADP and 2 inorganic phosphate to produce 2 ATP. So if we look at our inputs over here, we had the glucose in first, and that became 2 pyruvate. We've added our ADP and our phosphate, so our unloaded coenzyme there has picked up a phosphate and become 2 ATP. Now remember that this is respiration. The purpose is to make ATP. So some of them come in this stage. We also have, you notice in the inputs, NAD, which is another one of our coenzymes. So these hydrogen ions that were released when the glucose broke down, some of those are picked up by the NAD molecules, and they now become NADH. And we know that the job of these unloaded coenzymes is to move molecules around. So these NADH pick up the hydrogen, although they have become loaded acceptors when they have picked up their hydrogen. So the key take home from the process of glycolysis is that our glucose gets broken into molecules called pyruvates, and in this process 2 ATP are made. You need to know how many ATP are made in each section of respiration. So 2 are made during glycolysis. So if we go back to our summary diagram, we've done glycolysis. We're going to move into aerobic respiration now. So this will happen when there is oxygen available, which is the majority of the time. Remembering glycolysis occurred in the cytosol, whereas aerobic respiration occurs in mitochondria. So we're going to have a quick look at the structure of a mitochondria or a mitochondrion, similar to how we did with chloroplasts, because you do need to know which part of the mitochondria each section occurs at. So you will need to do a diagram like this in your notes if you haven't already. So mitochondria, you probably remember from year 11, we usually draw them kind of within our cell as little jelly bean shapes like this, often with a little squiggly line, okay? So these are our mitochondria here. A few key things to note. We have an outer membrane and an inner membrane. Similar to the chloroplast, we had two membranes there. There is a section in the middle, a kind of an opening in the middle called the matrix. Okay, and this is important because this is where the Krebs cycle, which is the first stage of aerobic respiration after glycolysis, this is where the Krebs cycle occurs. After that stage, the inner membrane, so this area here, is where the second stage, the electron transport chain, occurs. You see these words, the crista or cristae for plural, that just refers to the folds in the membrane. So I guess you could say that the crista is made up of membrane. A few other key things. We have ribosomes. So mitochondria have their own ribosomes. And if you think about what ribosomes do, that would give you an indication of what a mitochondria might be able to do. So I'll let you think about that for a second. Maybe even pause it. Yep, so having ribosomes indicates that proteins are made in a mitochondria. Remember, ribosomes being the site of protein synthesis. So that could include things like our coenzymes or even proteins that are found along the membrane. They would be produced in the mitochondria. Other key thing is that mitochondria have their own source of DNA. Small circular DNA. Again, this is very similar to a chloroplast. We actually call this DNA a small m and a small t DNA. That's worth writing down now. We'll actually come back to looking at mitochondrial DNA in Unit 4. It's really important when we look at things like evolution, surprisingly. Okay, so that is the structure of a mitochondria. Again, in the study design, VCAR wants you to know how mitochondria originally evolved from being bacteria. So we talked about this with chloroplasts, the endosymbiosis theory, which basically means that a bacteria was eaten by a cell, it entered the cell, and then they lived together. That's what endosymbiosis refers to. So the evidence we have of that is the size of mitochondria. They're quite similar to bacteria. So for organelles, they are quite large. They have their own DNA. So that mitochondrial DNA is similar to bacteria, have their own circular chromosome, and also these double membranes, which is quite similar to prokaryotes, which have a cell wall as well as a cell membrane. Okay, so that is our structure of a mitochondria, and we will move in now to looking at the actual steps that occur in aerobic respiration. I would perhaps maybe think about having a small break if you're going straight through with this now. Maybe look at the slides we've gone through, see if you can remember some of the key points, what happens in glycolysis. Can you maybe draw a mitochondria without looking at the picture? See if those steps have really sunk in before moving into the next bit. Okay, so if oxygen is available, the pyruvate molecules that were made in glycolysis are transported into the matrix of the mitochondria. Okay, that oxygen is needed to physically transport the pyruvate into the mitochondria. Again, I've got the pyruvate here in green because it is now an input of this stage. It was an output of glycolysis. It is an input of the Krebs cycle, which is what this section is called. The pyruvate molecules get broken down. So remember, they were 3 carbons initially. They get broken down. And as part of that, some carbon dioxide gets formed and is released. Okay, so you'll see our outputs here. We've got carbon dioxide as an output. We also get some hydrogen ions released. So remembering that the original glucose molecule had 12 hydrogens, some got released in glycolysis, a few more get released now. The hydrogens get picked up. We have some more of those NAD unloaded acceptors, unloaded coenzymes, and also another one called FAD. So they both pick up hydrogen ions and become NADH and FADH. Okay, so we've had our pyruvate getting broken down. We've got carbon dioxide getting made from those carbons. Our NAD and our FAD have picked up our hydrogens. The final thing that happens here, or the other important thing we need to know, is the ATP. Again, we're interested in how much ATP is made. So in this process, 2 ADP bind with 2 inorganic phosphate, and we get another 2 ATP made. So inputs here, outputs here. So we had 2 ATP in glycolysis. Now a further 2 ATP are produced in the Krebs cycle. These diagrams, there's a lot going on, and there's probably more detail than you need to know. But if you look at this section here with the circle here, VCAR love these. VCAR often have these kind of diagrams in their in their exam, and what they would generally do is they would cross out what these things are, and they might call this, let's say they might write molecule X, and you need to use your knowledge of the Krebs cycle and what goes in and what comes out to name what this is. So I wouldn't be spending too much time trying to memorize these diagrams, but you do need to know all of these steps. So what you should be doing is learning these steps and then using this picture to figure out what's going on. Because in an exam, it's not going to tell you probably wouldn't have this bit here. It wouldn't say that this is the Krebs cycle. You would have a diagram like that. It will tell you some of the inputs and outputs. So you'd know we're making ATP. So you'd be able to figure out we're talking about respiration. Hopefully, you would know that FAD and NAD are only used in respiration, that this is not about photosynthesis, and you would then need to be able to decipher, well, what isn't being shown here? Okay, maybe we're talking about the carbon dioxide that is produced as an output here. Okay, so these diagrams, I know they can look a bit confusing, but they do tend to show up in the exam a little bit. Okay, so Krebs cycle summary, the pyruvate came in. Now remember, 2 pyruvate were made in glycolysis. So this process will happen several times. So the pyruvate come in, they get broken down, some carbon dioxide is released, but we get 2 ATP produced in this process. So we're on 4 now from glycolysis and the Krebs cycle. The next stage is called the electron transport chain. This only happens if the Krebs cycle has happened first. Okay, so these are in steps. So remember, we've got our inputs in green here. We have the NADH and the FADH2. With this, each FAD unloaded coenzyme can pick up 2 hydrogen. So it's called FADH2. NADH and the FADH carry our hydrogen and electrons to the inner membrane of the mitochondria. So remember, the Krebs cycle happened in the matrix. Now the process actually moves into the membrane. Once those hydrogens and electrons are dropped off, they go back to being NAD and FAD. So if we go down now to our little graph here, we had input of NADH and FADH2. Once they get rid of their hydrogens, they go back to being NAD and FAD. Remember, once coenzymes pick up or regain their molecule, they can be used again. So they would go back and continue in the next stage. Now I've got a diagram on the next slide that shows this a little bit. You do need to know this word, cytochromes. Cytochromes are proteins that are found in the inner membrane. So remember, if you think of when we did the plasma membrane and we have our proteins all along a plasma membrane, it's the same thing here, okay? Even though we're inside a cell, it's the same principle. We know that organelles have a membrane around them. It would be a plasma membrane, and they have these proteins, okay? These are called cytochromes in the mitochondria. And these actually create a pathway. If you look back to the name of this stage, the electron transport chain, these cytochromes are producing that pathway for transport. Now, the electrons that were released by the NADH and FADH2 move along the chain, and they get picked up by oxygen atoms. Okay, so we had our oxygen came in. The oxygen will pick up electrons. And when they pick up those electrons, the oxygen becomes unstable. And you don't need to know too much about the chemistry around that, but unstable molecules bind with things easily. So the oxygen that has picked up electrons through the chain binds with the hydrogen that came from up here. So the hydrogen that the NAD and FADH released bind with the oxygen to produce H2O, produce water. Okay, so what you need to know about this is that the electrons move along the cytochromes and are picked up by oxygen, and then the oxygen binds with hydrogen to make water. So if we go over here, we had the oxygen input here, the output is hydrogen. Now you'll notice we haven't got an input of hydrogen here. Why is that? Because the hydrogens came in on our unloaded coenzymes. Okay, that's where the hydrogen has come from. Okay, so we've accounted for most of the things there, but again, the point of all of this is energy, ATP production. And this is where the money is. In the electron transport chain is where the majority of ATP come from. So every time two electrons move along this chain, 2 to 3 ATP molecules are made. Okay, so ATP is made by adding the phosphate to the ATP, as it always is. We get 2 to 3 ATP molecules for every 2 electrons. And this number here, this between 2 and 3, is where the variation in the equation for the total number of ATP comes from. So in total, during the electron transport chain, we get 32 to 34 ATP are made. Okay, you do need to know that. So we had 2 in glycolysis, we had 2 in the Krebs cycle, and then we have 32 to 34 produced in the electron transport chain, which gives us a total of 36 to 38 ATP, which is the number we use in our total equation. Okay, so that is where it all comes from. Again, we have another one of these diagrams here. It can look a little bit confusing, but really what's happening is we've got our NADH providing hydrogen, and we've got our FADH providing hydrogen here, and in this process we get ADP and phosphates combining to make ATP. We have our water output here as well. Like I said, I wouldn't be spending too much time trying to memorize this, but it can be useful to look through it now, knowing what you know about the electron transport chain, and seeing what you can decipher from that. This is a better diagram than what I drew of the cytochromes in the membrane, but if you actually look at this, you can see we have phospholipid molecules, same as a plasma membrane, and each of these is a protein. You don't need to know the names of these proteins, but they basically work as a mini production line, really, moving electrons along. This one is probably worth having a look at. Complex number 5 is ATP synthase. Remember, anything that ends in A's is an enzyme. Enzymes are proteins. So ATP synthase is the enzyme where ATP is formed by combining ADP with inorganic phosphate. Okay, so that is where that actually occurs. All right, I'll just give you a minute to have a read through this past exam question, and perhaps you pause it while you read through it, and then we'll go through it. Okay, so this question is asking which process produces the largest number of ATP molecules. Keep in mind in the exam, you're looking at a whole year's worth of content. So right now, this answer might seem quite obvious to you. But at the end of the year, it's really important that you know what each of these things mean. So A, the synthesis of polypeptide molecules. So that basically means during the process of making proteins. Now we've looked at this, protein synthesis, that does not produce ATP. That needs ATP, it needs energy, so it's not that. The breakdown of glucose during glycolysis, so we just looked at this, and this does produce ATP. 2 ATP are made in that process, so it could be that, so we'll write that. C, the light-independent reactions of photosynthesis. Now, if you went back and looked at your photosynthesis notes, you remember that in the light-dependent reaction, some ATP are produced by adding ADP and phosphate, and they are then used in the light-independent reactions. So it's not going to be this because no ATP is produced in that process. It's actually used. It goes back to ADP and phosphate. So that's not an option. D, the electron transport chain in cellular respiration, which we have just looked at, and we said that between 32 and 34 ATP are produced in this stage. So both B and D produce ATP, but the majority is made in the electron transport chain. Okay, so the process that produces the largest number of ATP molecules. This now is a short answer question. Again, I'm going to get you to pause this and have a little look at the question, and then we'll come back and work through it together. Okay, so we have the breakdown of glucose in aerobic respiration is represented by the simplified equation. So this is basically our word equation of respiration. First part, what is the energy yield per molecule of glucose as a result of aerobic respiration? So this term here, energy yield, is basically asking how many ATP are made. And this is through the whole process. Okay, we're not looking at one individual stage, it's in the whole process of aerobic respiration. And if you remember, for each molecule of glucose, sorry, I'm not sure where that went, each molecule of glucose, we end up getting 36 to 38 ATP. Okay, so that is your answer, 36 to 38 ATP. In the examiner's report for this year, I believe they accepted either 36 or 38, but just to be safe, if you know the equation, write 36 to 38 and make sure you write ATP. It's It's really important that you write that. You can't just write 36 to 38. The next part, the breakdown of glucose in aerobic respiration can also be represented as occurring in 3 particular stages. Okay, and we've just gone through these: glycolysis, the Krebs cycle, and the electron transport chain. The first question, within a cell, where does the electron transport stage occur? Now you need to be specific because both of these occur in the mitochondria. So let's be specific here. This occurs in the inner membrane of the mitochondria. Now notice it asked where in a cell. It didn't say where in the mitochondria, so you need to name both. Inner membrane of the mitochondrion. Part 2, describe what happens. So you need to explain a process here. Describe what happens during the electron transport stage. In your answer, include the name of product Z. So I'm going to give you a second to think about what you think product Z is. Remember, glucose came into glycolysis. What could product Z be? What is an output of the electron transport chain? Okay, now there's 2 options that this could be. One is water. Water is produced in that. The other is ATP. Okay, so either of those would be sufficient. So you need to specifically name what that is, and you need to describe what happens in this stage. So you could say electrons move along proteins and are picked up by oxygen. Oxygen bind to hydrogen to produce H2O, and then you could say in brackets product Z. Now there's a lot of things that happen in the electron transport chain. So you could either describe this process of the H2O getting produced, or you could describe the process of the ATP getting produced. So you could say every time electrons move along the electron transport chain, ATP synthase combines to ADP and 2-phosphate to produce 32 to 34 ATP total, and indicate that that is product Z. So narrow down what you want your answer to be and write your answer for that. Now we've looked at glycolysis, step 1. We've looked at aerobic respiration, so what happens when oxygen is available. Now we need to go through quite briefly this side of it as well. Okay, so when there is no oxygen available, what occurs then? The important thing to remember is that this still has happened first. So glycolysis happens first regardless of whether the process moves into aerobic or anaerobic respiration. Okay, so there's not enough oxygen available and anaerobic respiration occurs. Now this could happen either due to the environment that the organism is in, so maybe under the water, in a sealed or closed off area, high altitudes, for example, where there's not enough oxygen, or it could be due to increased demand, so doing really heavy exercise and not being able to take in oxygen as fast as you are using it. So glycolysis still happens. And remembering, in glycolysis, 2 ATP are produced. Okay, so that still occurs. Because there's no oxygen, the pyruvate that are made in glycolysis cannot move into the mitochondria. Okay, so they stay inside the cytosol. Now, it's the breaking down of the pyruvate in the Krebs cycle and then in the electron transport chain that results in the majority of ATP in aerobic respiration. Because this cannot happen, no more ATP gets made. So the 2 ATP that are produced during glycolysis are the only ATP that get made during anaerobic respiration because the pyruvate can't get broken down any further. We do get the production, as mentioned at the start, of these 2 byproducts. So in animals, this is our total equation, by the way, for the glycolysis and the anaerobic respiration. In animals, including in humans, the lactic acid buildup occurs in the cells, as well as those 2 ATP that were made here. And you may have experienced this before as cramps. That's actually what causes the cramping pain when you're doing strenuous exercise, is buildup of lactic acid when there's not enough oxygen available. And in yeast and in some bacteria, the glucose gets broken into the pyruvate, and then reactions occur that produce ethanol, which is the key ingredient in alcohols. So most alcohols are made by depriving certain grains or vegetables from oxygen, so it produces ethanol, and also carbon dioxide, which, if anyone's ever done baking with yeast, carbon dioxide is what makes yeast rise. Again, there's no mitochondria, they can't move into the mitochondria, it stays in the cytosol, only 2 ATP are produced in each of those. So it is a far less efficient process than aerobic respiration. Okay, I'm going to get you to pause here again and have a look at this exam question, have a good read through, and then we'll go through it together. Okay, so there's a fair bit of information in this, we'll work through it together. So yeast is a single-celled microscopic fungus. Sucrose is very similar to glucose. Okay, but you don't really need to know that. It's telling you what its food source is, and then it's going to explain, there's enough keywords in here that you'd be able to figure out what this is about. This is a really good example of a question that has incorporated an experiment. Like I said, that's a big focus on the exam now, that you need to demonstrate your knowledge of a topic, and often that's done through looking at experiments. So an experiment was carried out to investigate cellular respiration by a species of yeast. Okay, it's given you the topic here. Yeast cells were placed in a container and the sucrose, so the food source, was added, and an airtight lid was placed on the container. The measurements were taken of the percentage of oxygen and of ethanol in the container over a 1-hour period. Carried out at room temperature, so we know that that is our controlled variable. Okay, so the temperature was kept quite stable. So from here, it's told you we're talking about cellular respiration, and then it's told you that there's an airtight lid. So you should be able to start thinking we're looking at anaerobic respiration at a certain point. Now it's shown us here the percentage of oxygen and the percentage of ethanol in the container at the start of the experiment and at the end. So this was after 60 minutes, 1 hour. So at the beginning of the experiment, there is 21% oxygen, and by the end, there is 18% of oxygen. So the oxygen in the container has decreased from the start to the end. If we look at ethanol, at the beginning there is no ethanol in the container, and by the end that has increased and there is 4% ethanol. Okay, don't be afraid to write on questions in your exam. It will really help you clarify it. So This is a key word here. Explain any changes that have been observed in the oxygen and ethanol levels within the container. This is for 2 marks. This is a very big difference to if it said, for example, outline any changes. Okay. When it says explain the changes, you need to explain why this has happened. So you don't just need to say that the oxygen decreased. You need to explain why. And you basically want to break it into oxygen and ethanol. So you could say that oxygen levels decreased, and it doesn't hurt to put in the numbers, from 21% to 18%. Now, the reason this would be happening is if there was oxygen in there at the start, which there is, aerobic respiration will happen. Okay, so the yeast would go through some aerobic respiration while it can. So oxygen levels decreased as aerobic respiration occurred. And you'd need to explain why that means oxygen is being used. So aerobic respiration occurred, which requires oxygen as an input. Okay, so you're explaining why. You've mentioned aerobic respiration here. Now you need to explain why the ethanol has increased. So ethanol increased from 0 to 4% as anaerobic respiration occurred, which in yeasts produces ethanol as a byproduct. Okay, something along those lines. You need to reference the oxygen and the ethanol, and you need to reference the process that is happening. So either the aerobic or the anaerobic respiration. This is still the same question here, so take a minute to read through the this part of the question and then we'll discuss it. Okay, so this is now bringing in an extra component saying that carbon dioxide was also monitored during the experiment and to predict what would happen to carbon dioxide levels during that. So 2 marks, 1 mark is going to be for the correct prediction and 1 mark for the explanation. So if you remember our equation for alcohol fermentation, which is what occurs in yeasts and plants, so glucose becomes ethanol and carbon dioxide and our 2 ATP. So we saw the increase in ethanol. Now it's asking about carbon dioxide. So you would predict that CO2 would increase. Hasn't asked you to give any number. You're not expected to know by how much. And you could say that CO2 is a byproduct or an output of anaerobic respiration once the oxygen was used and anaerobic respiration occurred, CO2 would be produced along with ethanol. Okay, so explaining the reasoning behind your prediction is very important. Now, with all of the topics we've looked at things that affect the rate at which these processes occur. Again, these could easily come up in your exam, where they could, do a pretend experiment, for example, and then ask you, what could be the impact of doing these things. So they are important. So similar to photosynthesis, we look at the limiting factors, or the factors that affect the rate, I guess, is what you're looking at here. And you'll find that these two are both inputs, okay? These are inputs of respiration. Without glucose, respiration is not going to happen. It's the primary input. And remember, with oxygen, we're looking here at our difference between aerobic and anaerobic. Okay, so the rate of respiration is going to increase the more oxygen is available. Temperature, again, we're looking at the impact on the enzymes. So the enzymes will kind of increase their efficiency up until their optimal point. And then remember, if they bypass their critical point, the process will stop. So it's important that you understand what this looks like in terms of graphs. So with glucose, if we don't have any glucose, we don't have any respiration. If more glucose becomes available, the rate will increase until it gets to a point of leveling out, because maybe other things at that point are limiting it. So maybe all of the mitochondria are in use, or all of the enzymes, so the NAD and FAD, all of them are being used. Oxygen concentration, same thing. So to an extent, the more oxygen we have, the faster respiration will happen, until it gets to a certain point where it can't go any faster. So it levels out. And then our real difference is the temperature curve. So remember, as we approach our optimal temperature, which is probably around here, as we approach that, the rate increases, and then we'll get to a certain point, probably around here, where it's the critical temperature, and then the enzymes at that point will denature. So remember, the proteins will change their shape. Let's say, for example, our FAD now changes shape and cannot pick up the hydrogen anymore. Okay, so they're examples of when these things will stop working and the process will eventually stop. So note that difference between the different limiting factors. Okay, we're going to finish off by looking at photosynthesis and respiration together. This definitely comes up in the exam a lot. Can you tell the difference between each process? And can you also really analyze and describe the relationships between the processes? So we'll look at a few past exam questions and also some graphs that really show this quite well. Okay, so I'll give you a bit of time just to read through this first question, where a student was asked to identify differences between the overall processes of photosynthesis and aerobic respiration. And then the question is asking you to identify the only correct comparison. I'll give you a second to read through that, maybe pause it, and then we'll go through it. Okay, so if we read through these one by one. So for photosynthesis, they have written that photosynthesis is exergonic. Remember, exergonic means releases energy, and endogenic means that it uses energy. And that is the wrong way around. Photosynthesis is endogenic, it uses energy, while aerobic respiration is exergonic because it produces or releases energy. So this is false, and we have to identify the only correct comparison. Photosynthesis, all stages occur within chloroplasts. Aerobic respiration, all stages occur within the mitochondria. Okay, so in photosynthesis, both of our stages do occur in the chloroplast. In aerobic respiration, it begins in the cytosol. Okay, so glycolysis happens in the cytosol, which is not in the mitochondria. So that is incorrect. Photosynthesis and aerobic respiration. So photosynthesis, electron transport is not involved. Aerobic respiration, electron transport is involved. Now we know this is true. Even though in photosynthesis we don't have a stage called the electron transport stage, if you think back to the NADP, which picks up hydrogen, it also picks up electrons. So NADPH transports electrons in photosynthesis. So even though it's not its own stage, there is the transport of electrons in photosynthesis. So this is not correct. We'll look at this final one. Photosynthesis uses water as a reactant. So remember, a reactant is an input in the first stage. And the first stage of photosynthesis is the light-dependent stage, where sunlight energy splits the water into H2O. So we know that this bit is correct. And then aerobic respiration forms water as a product, so as an output in the final stage. And we know this is correct. In the electron transport chain, hydrogens and oxygens bind together to produce water. Okay, so the correct answer here is D. Question 8. Again, I'll give you a minute to have a read through. Please pause this as you read through, and then we will work through it together. Okay, there's a fair amount going on in this picture. This is just a bit of introduction. So climate change has been linked to an increase in CO2 in the atmosphere. And then it's talking about small algae, so microalgae, can use greater amounts of carbon dioxide than land plants and do not require prime soil, reliable rainfall, and a particular climate. The question goes on, and we're not going to look at all of it. The question goes on basically to look at whether chlorella is a suitable organism to be used in farming and production because of how it works. But I just want us to focus on the relationship here between photosynthesis and respiration. Now, let's take a minute to actually annotate this picture, because it'll help you narrow down what's going on. So it says here that M, N, O, and P are processes. Okay, so they're not structures, they are processes that are occurring. So what we want to do is figure out which ones they are. So it says we've got an input here of sunlight. And the only process that we've talked about where sunlight is an input is the light-dependent stage of photosynthesis. Okay, the light-dependent stage. Now, there is a compound that is released from this that goes into here, and then we also have a relationship over here. Now, I think let's look at our keywords here. Glucose, whatever stage N is, produces glucose. So there's a relationship between M and N, and then N produces glucose. We've also got the information here that carbon dioxide is an input into stage N. So we could say that N now is our light independent stage of photosynthesis. Okay, so the ATP and hydrogen from the light dependent stage travels over to the light independent stage. Carbon dioxide diffuses through the atmosphere into the plant, and glucose is produced there. Okay, so then we know that glucose is an input for process O, and compound Y, which we'll come back to, is an input for process P. So we know that glucose goes into our cellular respiration. Now this has told us here that between process O and P, pyruvate is made. So O must be glycolysis, and then P would be aerobic respiration. Okay, so the pyruvate enters aerobic respiration. What would be our compound that is released from the light-dependent stage of photosynthesis and enters aerobic respiration? I think the aerobic there is the key word. That compound Y would be oxygen. Remember, oxygen is made in the first stage of photosynthesis and is released, and then it enters here. Okay, so the only other thing we haven't really identified here is what input X is. So we've got the sunlight energy. So sunlight's not technically a molecular input, and it's already given us the sunlight here. So our other inputs of the light dependent stage, it's not carbon dioxide, that comes in the light independent stage. So our input X would have to be water, or possibly ADP. There is some ADP, but I think let's stick to the ones that are in our equation. So input X is water. So you could add input X there, water. Feel free to put the symbol if you'd like. And then compound Y is oxygen. Okay, don't worry too much about the amounts. It's just asked you to name it. Okay, this question continues on. I have put the picture back there for us to look at. Now, with reference to the diagram above, complete the following table. Now, we had already annotated this. So, process M was the light-dependent stage. And I think it's worth writing of photosynthesis. Okay, now it's given you the site for that. We know that the light-dependent stage occurs in the grana or in the thylakoid of chloroplasts. Now for section O, which is glycolysis, it is given us the name. We need to say where that occurs. So glycolysis occurs in the cytosol of the cell. Okay, and then P is the stages of cellular respiration. And it's specifically the stages of aerobic respiration because we know oxygen has come in. So therefore, our site for that is a mitochondrion. Okay, I really like this diagram. I think that is quite similar to some of the ones we've looked at. You might even play around with that, and you could use something like that as a bit of a summary diagram for you. This is a bit more full on. I'm not going to go through this too much. This is just a really good option for people that are very visual learners and really like to see the relationships between everything that's occurring. So we have the photosynthesis on the left and the respiration on the right, and it kind of summarizes the key processes that occur. So you may like this particular diagram, or you might want to do something similar to this when you are doing your summary and revision tools for this. This is something you need to know. So often in the exam, the rates of cellular respiration and photosynthesis will be mapped together. When you see things like this, let's assume that we are looking here at a particular plant. And the reason we should do that is because plants go through both photosynthesis and cellular respiration. Okay, we go through cellular respiration, but our source of glucose is through eating, not through photosynthesis. So let's assume this is a plant that is doing both of these processes. This is saying the rate of the process, and then this particular diagram or this particular graph is looking at the change in the rate based on the change in the amount of light. So you can see here that the respiration stays the same. When we're only comparing it to light intensity, light does not impact the rate of respiration because light is not, it's not an input, it's not an energy source for respiration. But you can see there's quite a big difference in the rate of photosynthesis. So if we're saying this is when there's not much light, so it is dark, so it might be at night time, the rate is quite low. And then at point C, the rates are equal. So the rate of photosynthesis and the rate of cellular respiration is equal. And I guess what you could say at that point is that the same amount of glucose is being used as is being produced. And the same for oxygen and for carbon dioxide. If we were to, depending on what this unit process is, whatever the input or output we're talking about here is equal. And then as it gets really bright, so let's say it's the middle of a bright sunny afternoon, the rate of photosynthesis is actually higher now than respiration, which means there's probably more glucose. And what is our other output of photosynthesis? We have our oxygen. So because photosynthesis is happening at a higher rate than respiration, there would be more glucose and oxygen in the plant than there would be at this process when that is getting used at a greater rate. So this is looking at a plant over 24 hours and looking at the rates of photosynthesis in green and respiration in red. Give you a little minute just to look through that and to have a look at that question on the left. Please pause it while you go through each step and then we'll come back. Okay, so question 1, what accounts for the difference in the rates of photosynthesis and cellular respiration? So what accounts for the difference between this line and this line? And hopefully these times give you a bit of an indication. It is the light. Okay, so the amount of light particularly impacts the rate of photosynthesis. Much bigger variation in the rates of photosynthesis than there are of respiration, really dependent on the light. When it is dark, there is almost no respiration happening. In the afternoon, it's at its highest rate. Okay, now this question might take a minute or so to figure out. So at each of the following points, so A, C, and E, is the plant taking in or releasing more oxygen? So think for a second, which of these processes, is it photosynthesis or respiration that releases oxygen as a byproduct? Pause for a sec if you need. Okay, so it is the process of photosynthesis that gives off oxygen. Whereas respiration consumes oxygen. For respiration. Okay, anytime photosynthesis is occurring more than respiration, oxygen is going to be released from the plant. Okay, so if we start with C, C is probably the easiest one. At C, the rate of respiration and the rate of photosynthesis is equal. Okay, so that means any oxygen that is made in photosynthesis is getting used by cellular respiration at the same rate. So at point C, you would say there is no net intake or release. Or you could even just say that the rates are equal. Okay. Let's look at point A. So point A, just after midnight, no photosynthesis is occurring, but respiration is occurring. Okay, now we need oxygen for respiration, and it's not being made by photosynthesis. So where is that oxygen coming from? It's having to be taken in. Okay, so in point A, the plant is taking in oxygen because it needs oxygen for this process and it's not able to produce it in photosynthesis. At point E, we're back over this line now, so photosynthesis is occurring at a higher rate than respiration. Some of the oxygen gets used by respiration, but the additional oxygen gets released by the plant. So it goes out into the atmosphere. Okay? Give you a minute again to read through this question, and then we'll have a look at the questions on the back. Okay, so again, another example of VCAR using an example experiment to assess understanding. So it's basically saying we're looking at the study of aerobic respiration and 3 different setups were used. One of them contained just mitochondria. One contained just cytosol with no mitochondria. And one contained mitochondria and cytosol of cells. You'll see why this is really important. Here we've got our controlled variables, and then the important information here is what was added to each of these 3 tubes. So in the first one, glucose was added to the mixture, and then the oxygen concentration was measured. Now, let's just think about this for a second. If respiration is occurring, what should we see happen to oxygen levels? I need to think for a second. Okay, if respiration is occurring, oxygen levels should decrease, because remember, oxygen is an input for cellular respiration. In each experiment, glucose was added to observe what happens to the oxygen, and then pyruvate was added, and again the oxygen was measured. Okay. Okay, so the actual questions here now ask you again to make some predictions and justify those. So it has said to use your knowledge and understanding of aerobic respiration and mitochondria to predict the change in the oxygen concentration, and then explain why. So in the first experiment, we are looking at the tube that just has mitochondria, doesn't have cytosol. Okay, so if glucose was added to this, so glucose was added to mitochondria, what do you think would happen? Pause for a sec if you'd like to. Okay, if we add glucose to mitochondria, respiration won't happen because mitochondria accept pyruvate and break down pyruvate. Glucose has to be broken down in the cytosol. So there would be no change in the concentration of oxygen because respiration wouldn't occur. Okay, we can do the explanation in a second. Whereas if pyruvate were added, so remember pyruvate go into mitochondria, into the matrix, and that's when the Krebs cycle and then the electron transport chain occurs. So if pyruvate enter the mitochondria, they'll go through the Krebs cycle, and then they will move into the electron transport chain. And this process will use oxygen, so there would be a decrease in oxygen concentration if pyruvate alone was added to a mitochondria. Okay, so you could say here that pyruvate are broken down in the mitochondria during aerobic respiration, which uses oxygen. Okay, whereas for glucose in mitochondria, glucose must be broken down in the cytosol before aerobic respiration can occur. So therefore, no oxygen will be used as aerobic respiration isn't occurring. Okay, and then this is almost the opposite down here. So experiment 2, where it's just the cytosol with no mitochondria. So this time, if we put glucose in the cytosol, I want you to think about what would happen and would that impact oxygen concentration. So just pause for a second. Now, if we think about it, glucose, if added to mitochondria, will get broken into pyruvate. We know that. Now, if there's no mitochondria here, will step 2 occur? So if there's no mitochondria, we're not going to have aerobic respiration occur. So this would remain in anaerobic respiration because there's nowhere for the pyruvate to actually move. So again, there should be no change in the oxygen concentration. And again, with pyruvate will remain in the mitochondria, in the cytosol. Again, anaerobic respiration will continue, so we might get an increase in other byproducts. So it could be ethanol, it could be carbon dioxide, but we're not going to get any oxygen being used because aerobic respiration cannot occur. So for both of these, no mitochondria for aerobic respiration to occur, so no oxygen is used. And it would be a similar thing for both. You could, to demonstrate your understanding, you could explain in this one that the glucose would still be broken into pyruvate. Here you could explicitly say that the pyruvate cannot transport to the mitochondria as they're not there. So aerobic respiration cannot happen. Okay, so that is the conclusion of this PowerPoint. As I said, I really hope you've done this over a few sessions. If not, I recommend spending a bit of time going through this. There's a lot of information in there, and it's best just to consolidate little bits at a time. So we did go through the purpose of respiration. So that is producing energy in the form of ATP by breaking down glucose, the location, and the inputs and outputs of glycolysis, which is the process that occurs in the cytosol, and we have our 2 ATP that are produced from that. We've looked at the structure of mitochondria as the site of aerobic respiration, And remember, we looked at the bacterial origins, so the things like their own DNA and ribosomes, and the double membranes and their size. Then we've also looked at the inputs and outputs of the Krebs cycle and the electron transport chain. So they were our second two steps of aerobic respiration, including the ATP yield. So we get the 2 ATP in Krebs cycle and 32 to 34 in the electron transport chain. We've looked at the location of anaerobic respiration, so that occurring just in the cytosol, and the difference in the outputs between animal and yeast. So again, we looked at the ATP, just the 2 ATP, but also the outputs including the lactic acid, the ethanol and carbon dioxide. And then finally, we had a look at the factors that affect the rate of respiration. So the temperature, glucose availability and oxygen concentration. That is it for this session. As I said, please have a look through the Ed Rollo and your textbook. There's lots of practice questions in your textbook, so make sure you work through those. If there are any sections of this that you want more information about or really didn't make sense, please send me an email. But we'll be doing a Zoom in our first week. If we're not back at school, we will jump online and start talking through it then. So if you've watched this now, it's probably a good idea to have a look through again just before school goes back. All right, take care and I'll chat to you all soon.